Right, this is my video on Unit 5.6 Photosynthesis. If you've not watched respiration, I recommend you do watch it. So you need to get a grasp of oxidative phosphorylation because it is really key, as it links so much to photophosphorylation in photosynthesis. And if you understand respiration, it's likely you'll pick up photosynthesis pretty quickly. Right, anyway, so this is the equation for photosynthesis. The two molecules we're using here is CO2 and water, H2O, producing glucose and oxygen. So that is the balanced chemical equation, which you might want to learn and write down. So the main aim in photosynthesis is here on the left. So we've got photons, which are particles of light, and each photon contains a quantum of energy. So photosynthesis is an example of carbon fixation, the process of converting carbon dioxide into sugars. It is an endothermic reaction. I've not written that down. Addition of electrons makes it a reduction reaction, but also there is elements of an oxidation reaction. So reduction and oxidation, they come hand in hand together, really. And our products, so glucose and oxygen, they go into respiration. Okay. Let's have a look at where photosynthesis takes place. So it takes place in the organelles called chloroplasts. They've got a double membrane with an outer membrane, intermembrane space, and then our inner membrane. Within that membrane, we've got the stroma, which is where the light-independent stage takes place. That's the fluid-filled matrix. It contains the enzymes needed to catalyze light-independent photosynthesis stage. So it's got starch grains, oil droplets, small ribosomes, and DNA in it, which produces proteins needed in respiration and enzymes that I've already mentioned. Then we've got these grana, which is these stacks. Here's a 3D image of it. Stacks of thylakoids. That's where the light-dependent stage of photosynthesis takes place. Thylakoids within granum are connected to other thylakoids within other granum by intergranal lamellae, which are these lines here in a 3D image. They're here and here. Thylakoid membranes are less permeable because they're just like oxidative phosphorylation. They're containing hydrogen within the intermembrane space. One granum can contain up to 100 thylakoid stacks, so lots of grana create a huge surface area for distribution of photosystems, which I'll speak to you about in a minute, and electron carriers, which I'll also speak to you about in a lot more detail soon. One last thing to look at on this page is this diagram here, talking about compensation point. So plants respire all the time. This is the line of respiration on the diagram. By the way, on the left we've got O2 delivered, and at the bottom we've got the time of day. So this would be the peak of the day when we've got the most sunlight, and that's either end, sunrise, sunset, etc. Alright, so plants respire all the time, but they only photosynthesize during daylight. So plants have interspecific competition for light. So here we've got green, the shade plant, and here's a sun plant. The light intensity must be sufficient to allow photosynthesis. So shade plants can deal with a lower intensity of light. That's how they're adapted. Sun plants require, as you can see, in the middle of the day, they need really high intensities of light for photosynthesis. When photosynthesis and respiration are at the same rate, it's called compensation point. So these are the blue lines. So photosynthesis at these blue points here circled is at the same rate of respiration. So there's no net gain of carbohydrates. Shade plants can utilize lower intensity of light than sun plants. When exposed to light after being into darkness, they can reach compensation points sooner and have a smaller compensation period. So shade plants are better adapted to get out of this compensation point. Some plants really need optimal light intensity. Okay, so now let's have a look at photosystems. So photosystems of different wavelengths, the light hits them, it gets absorbed. The energy from that light, from those photons of light, is transferred to this primary pigment reaction center, which contains chlorophyll A. Okay. I've spoken about all of that. So that primary pigment reaction center is at the base, so it filters down all the energy into here. Now let's have a look at our chlorophylls and accessory pigments. So chlorophyll can be A or B. Chlorophyll A is always blue or green, so it refracts blue and green light. It is at the center of photosystems, so it's found in this primary reaction center that is all containing chlorophyll A molecules. Okay, so chlorophyll A can either be P680, so an optimal wavelength of 680 nanometers, and that is found in photosystem 2. So there's two photosystems, photosystem 1 and 2. P720, which has an optimal wavelength of 720 nanometers, is found in Photosystem One, so PS1. Please in the exam write down Photosystem One, then you can put in brackets PS1, just to make it, just to guarantee you're going to get the mark, because you never know. You're at PSII, they might not give you the mark. So write down the full name. I've just done it as note form here. But both photosystems absorb some blue light, which is around 440 nanometers. Then we have chlorophyll B, which is yellow and green, so it refracts yellow and green light, and its optimal wavelength is between 400 and 500 nanometers. Then we have accessory pigments. So accessory pigments, like chlorophyll B, which will be found in here, accessory pigments are also found here. So there's two I've written down here. There's probably more. You might need to see if you need to know any more, but these are the two I'm learning, and the only two I'm going to learn. Carotenoids, which absorb a light wavelength between 400 and 500 nanometers, and xanthophylls, which absorb a light between 375 and 550 nanometers. So chlorophyll B and accessory pigments are found in this bit here. They absorb light, and then they filter that energy down into chlorophyll A, which can either be P680 or P720, depending on the photosystem. So these are our two photosystems, Photosystem 1 and Photosystem 2. Photosystem 2, as well as having P680, it also undergoes a process of photolysis. So photolysis is the splitting of water molecules. So this is the equation for it. 2H2O goes to 4E minus plus 4H plus, so 4 electrons, 4 protons, plus 2 O2. Photolysis is the sort of all protons in photophosphorylation, which I've written diagonally here in a really poor handwriting, which is the stage I'm just about to describe to you. So that these are photosystems. They have a huge role in the light-dependent stage of photosynthesis to produce ATP for the Calvin cycle to function. Okay, this is our light-dependent stage. Also, photophosphorylation. There's two forms of photophosphorylation, non-cyclic and cyclic. Once again in the exam, when you write these words, write non-cyclic photophosphorylation and cyclic photophosphorylation, not just cyclic or non-cyclic on its own. Okay? I'm going to explain to you first non-cyclic. It is the main form of ATP production in plants. It's quite complicated, but you will also see it's got a very similar resemblance to oxidative phosphorylation within respiration. So first of all, a photon of light strikes PS1, so photosystem 1. So we have light strike PS1. Okay, I'll draw these wavelengths. That's light hitting it. Okay, let's get rid of that now because I don't have too many lines on this diagram. So light hits this. That excites a pair of electrons. Okay? So the light energy hitting photosystem 1 excites a pair of electrons within the chlorophyll A at the bottom, okay, in the primary pigment reaction center. Okay? So the light is absorbed, its energy excites a pair of electrons in the chlorophyll molecules. The energized electrons then escape from the chlorophyll molecule. Okay, so I'm going to draw that on for you. In respiration, I did them in blue. I'll do that again. So PS1, photosystem 1, releases 2E minus. Okay, it releases a pair of electrons into our matrix. But straight away, by the way, we're looking here. We've got inner membrane, intermembrane space, outer membrane, just like in respiration. So these two electrons then are taken up by this protein carrier. So once again, it's an ion, Fe2+. It's a protein with an ion in the ion in the center. So this electron, these electrons are taken up by Fe2+. This causes Fe2+ to be reduced. And what do we know about being reduced? Yeah, that means you get in a hydrogen or electron in this case. So instead of 2+, it becomes 3+. Okay, so it's taken on board this electron. It's become reduced. Photosystem One has become oxidized as it's lost the electrons, and now this ion in the protein carrier has been reduced. Okay, this keeps happening, so it keeps passing the electron between these ion carriers, okay, embedded in protein molecules. So when that happens, we're seeing a constant cycle of reduction and oxidation. So when this ion passes on the electron to the next one, it becomes oxidized. So once again, let's change that back to 2+. 2+, because it gets rid of the electron and it transfers it to the next ion, so this ion becomes reduced, so it goes to 3+. Same thing happens again now. That becomes oxidized by releasing this electron to here. That becomes 3+. So that this one is now reduced, and this one oxidizes itself by releasing the electron. Okay, so hopefully you understand what I'm talking about here. The electrons are passed down these electron carrier proteins, constantly oxidizing, reducing each other, until we get to photosystem 2. So the electrons passed on to photosystem 2, which we said contains P680 of chlorophyll A. Therefore this last ion therefore gets oxidized, and the photosystem 2 becomes reduced. So as we said earlier, well, you can't see me zooming out, but I am. That's how I can write fairly small on this, because I can zoom in and out. So photosystem 2 undergoes a process called photolysis. So it splits water into H+ ions, which I'll draw in this sort of colour. So H+ is released, 4H+ from water. I'll write water in photosystem 2, H2O. So that is split up. I'll draw a cross through that if you saw what I just did there. H2O, it's split up into our hydrogens and electrons. I don't want that colour actually, we want blue for electrons. And electrons, 4E-, and of course our oxygen, which is a byproduct. And it goes into the stroma, okay, so O2. Right, so we've got hydrogens and electrons released by photolysis. The release of these electrons allow it to have capacity to take on these electrons from the ions, which has come originally from photosystem 1. So electrons are carried along these electron carriers up to photosystem 2. Electrons move across ions. Electrons carry a lot of energy, so when they move across, it releases energy. Let's draw a little yellow flash to symbolise energy. Okay, so energy is being released as electrons pass through it. And what do we know about that? Just like an oxidative phosphorylation, it allows the H+ to be pumped into our intermembrane space. So H+ is now accumulating in our intermembrane space. Remember, all these hydrogen ions or protons have come from the photolysis of water, the splitting of water. Right, so they accumulate here, they form a high chemoosmotic potential, just like in respiration, and here is our ATP synthase molecule. Once the concentration of hydrogen is high enough and the chemoosmotic potential is big enough, the hydrogen ions are pumped through this ATP synthase molecule. So the chemoosmotic potential causes them to be pumped through. What do we know that does? It turns the heads here, let's colour them in. These heads are turned to cause ADP plus P to go to ATP. Let's do ATP in pink. ATP. And then ATP will then also enter the stroma. So that is our main product, ATP, produced with here. And now we've got an issue again. We've got hydrogen ions that have just been pumped through, accumulating here. No, wrong colour. I've got so many colours going on here. Hydrogen ions pump through to produce ATP. They accumulate there, and that's when we've got this NADP. So NADP accepts, that's right, NADP accepts our protons and electrons. So NADP, when it accepts protons and electrons, it becomes reduced. So we put a H on the end. So that is reduced NADP. Okay, NADP enters the stroma. Okay, let's get rid of that arrow because there's one more thing I've got to tell you about, and that is Ferredoxin in Photosystem One. So Photosystem One contains a protein iron sulfur complex called Ferredoxin. It accepts electrons from Photosystem One, which has been passed to it from these carrier proteins from the left that I've not drawn on, and passes them to NADP in the stroma. So not only do electrons come from Photosystem 2, they're also coming from Photosystem 1, and they're passed to NADP in the stroma. So then we've got this equation here, so NADP plus E minus plus H plus, so protons and electrons and NADP, by the catalyst NADP reductase forms reduced NADPH, so reduced NADP. There are two products then, so we've got NADP, ATP, and of course oxygen, which is our byproduct. So that is non-cyclic photophosphorylation. Lots of similar things there to oxidative phosphorylation, but obviously a few more. I'd say it's a bit more complicated than oxidative phosphorylation, if that wasn't complicated enough as it was. If you didn't understand that, try and watch that bit of the video again. It is quite hard, especially if you struggle with respiration. Also, probably just do a bit more background reading on it, and eventually you will understand it. Cyclic photophosphorylation is the second form of the light-independent stage. It produces much less ATP, a much lower yield of ATP, so it's not always happening. But in guard cells, they only have Photosystem 1, so they can only carry out cyclic. So cyclic photophosphorylation is when electrons are passed between Photosystem 1 electron carriers, which are Fe2+, and back to Photosystem 1s. This movement of electrons between Photosystem 1 and another Photosystem 1 across our protein carriers with ions within them, that creates energy. That energy can be used to generate a small amount of ATP. So we get no NADP produced, no protons, no photolysis of water. An example of cells that use this are guard cells. They only have Photosystem 1 cells. They use only cyclic photophosphorylation. They've got no need for the Calvin cycle, which is the light-independent stage, which I'm going on to now to simplify it down. So 6 RUBP, ribulose bisphosphate, combines with 6 carbon monoxide. Through action of Rubisco enzyme, this forms an unstable 6-carbon compound, which immediately splits into 2 GP. But because we had 6 IPP and 6 O2, we get 12, 6 CO2, sorry, we get 12 GP. Okay? 12 GP are each phosphorylated, so we get 12 ATP to ADP plus P. They're all phosphorylated with one phosphate group each. They are all reduced with 1 hydrogen each by action of NADPH, reduced NAD to oxidized NADP, which can then re-enter the light-dependent stage. So this phosphorylation, I'm going to write that down, it's not on the diagram, phosphorylation and reduction forms TP, triose phosphate. TP is our product molecule that we want. It's got 12 TP produced. This is used for synthesis of amino acids, fatty acids, and glycerol, and converts glucose to sucrose, starch, and cellulose. Okay. 10 of our 12 triose phosphate are used to resynthesize RUBP. So only 2 product molecules are produced, which is quite disappointing, really. It's not a high yield, but this cycle can happen loads of times, and we're constantly producing a small amount of TP, or a large amount of TP, but only a small amount is actually used as product. The rest is regenerated here, or resynthesized into RUBP by phosphorylation. Okay, so triose phosphate's recycled. That is the Calvin cycle. I understand my diagram was not that useful to you. It took me a lot of cuts to explain this, so sorry for this. That was really fragmented for you to watch. I will be editing it afterwards though. Finally, we've got our factors affecting photosynthesis. So we've got 4 factors here. First of all, we've got light intensity. So of course, light intensity is going to affect photosynthesis the most out of anything. First of all, because light is essential for the light dependent stage. It carries our photon energy for the light dependent stage. Presence of light also opens stomata for gaseous exchange. When light intensity is low, photosynthesis rate is low. So if there's little or no light, we cannot reduce GP into TP because NADPH is needed from NADPH, reduced NADP, is produced from the light dependent stage. If we have no light, the light dependent stage can't happen. We can't produce reduced NADP, and therefore we cannot reduce GP to TP. So if you don't get light, first of all you get no ATP, and then you get no TP. It has a knock-on effect. Second factor is CO2 concentration. Usually it's very rare to affect it, as there's high enough levels in the atmosphere that CO2 is not usually a limiting factor. However, CO2 below 0.01% causes RUBP to not be able to accept it. GP cannot be made as RUBP is accumulating. There's no CO2 for it to bind to. Here, RUBP, if there's no CO2, you can't make GP, then you can't make TP, of course. Okay, temperature. When all other substances are available, temperature becomes the limiting. All of the substances become available. So if we've got high light intensity and high CO2 concentration, temperature becomes our limiting factor. Temperatures between 25 and 30 degrees are optimal. As the temperature increases, so does the rate of photosynthesis. But temperatures above 30 degrees, growth rates reduce due to photorespiration. O2 competes with CO2 for enzyme rubisco, so CO2 can't bind with rubisco, therefore GP cannot be produced and TP cannot be produced. That is a common link here we're seeing with factors affecting photosynthesis. You've got to look at what they're going to block. It's usually this reaction is blocked. So when we've got no CO2, we can't produce GP, then we can't produce TP. If we've got no light, we can't produce NADP, reduced NADP, and then therefore we can't reduce GP into TP, etc., etc. Okay, it's a common thing. Our final one is water availability. So we know water is used in hydrolysis, so it provides photolysis, so it provides hydrogens and electrons. If we've got no water, we cannot produce ATP, we cannot produce NADPH, nothing is produced. So water really is an essential element. However, you don't need too much water for photosynthesis to be able to occur. Our final thing to look at now is this piece of apparatus here, really badly drawn. It's a photosynthesizer. Here I've got a thermometer. I've got some pondweed within a conical flask underwater. And basically we're measuring the oxygen produced by this pondweed. And you can vary the light intensity on it, wavelengths of light by having different coloured acetate around it. The temperature, as we see here, so we could vary the temperature and see what rate that effect that has on photosynthesis. We can also add sodium hydrogen carbonate solution, varying the amount of this in the solution. So this syringe here, we can either measure the movement of the meniscus, or we can just straight off measure the amount of oxygen produced in here. However, there are some limiting factors. Some oxygen produced is respired. You need the equipment to be airtight. There should be no air bubbles. You set it up underwater. And also there's gases such as nitrogen and carbon dioxide can also be released. It's not all oxygen. It's not actually a representation of how much photosynthesis is taking place. So that was unit 5.6 on photosynthesis. A tricky unit, but honestly I understand it really well now. You just have to keep looking over all these processes, especially photophosphorylation. And if you understand oxidative phosphorylation, you should quickly get a grasp of this. Okay, understand what photosystems are, understand oxidation reduction. You should know how ATP synthase works. If I've not explained this well enough as it is, go and have a look at some different sources and try and understand what the Calvin cycle is. Okay, there is only really 3 or 4 steps to it to remember, and while it looks complicated, you've got Rubisco, GP, TP, IBP, Rubisco, GP, TP, and of course CO2, and that's all you really need to remember, and just the steps in between that. So it's quite a messy diagram. Try and find your own one if you don't understand it, but thank you, that's all I'm saying today.